Welcome to the world of molecular technology. I'm Dr.A. Jayasree, Department of Microbiology, Nehru Arts and Science College. In this fifth semester, you will be learning Recombinant DNA Technology 1 as your elective paper 1. The course objective is to make you knowledgeable on the gene manipulation techniques and tools used for construction of gene cloning. And also, you will be able to understand about the construction of transgenic plants and animals. Course outcome. On completion of course, the students will be able to understand about the gene cloning and tools such as restriction endonucleases, DNA modifiers, know about plasmids and their derivatives, know about different gene transfer methods, screening and selection of recombinant DNA, and gene analysis methods. Know about the application of genetic engineering for construction of transgenic and microbial products. Gain knowledge about the applications of interferons and growth hormones. Unit 1. In this unit, you will learn about what is recombinant DNA technology and gene manipulation methods, isolation and purification of DNA, RNA, and plasmids, restriction enzymes such as exonucleases and endonucleases, its types and mode of action. DNA modifying enzymes such as ligases, methylases, phosphokinases, and their mode of action. Recombinant DNA technology. According to the definition given in the Encyclopedia Britannica, rDNA technology is the technology used to join together DNA molecules from different organisms and inserting it into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. It is also known as genetic engineering. The technology was developed after years of research by Stanley Cohen and Herbert Boyer in 1973. It led to rapid and efficient DNA sequencing techniques that enabled the structures of individual genes to be determined. What is gene cloning? Gene cloning, also known as molecular cloning, refers to the process of isolating a DNA sequence of interest for the purpose of making multiple copies of it. The identical copies are clones. Let us have a look at the basic steps in gene cloning. First step, construction of a recombinant DNA molecule. A fragment of DNA containing the gene to be cloned is inserted into a circular DNA molecule called a vector to produce a recombinant DNA molecule. Second step, transport into host cell. The vector transports the gene into a host cell, which is usually a bacterium, although other types of living cells can also be used. Third step, multiplication of recombinant DNA molecule. The vector multiplies within the host cell, producing numerous identical copies not only of itself but also of the gene it carries. Fourth step, division of host cell. When the host cell divides, copies of a recombinant DNA molecule are passed to the progeny and further vector replication takes place. Fifth step, numerous cell division resulting in a clone. After a large number of cell division, a colony or clone of identical host cell is produced. Each cell in the clone contains one or more copies of recombinant DNA molecule. The gene carried by the recombinant molecule is now said to be cloned. What is PCR? Polymerase chain reaction is a technique of amplification of specific DNA region in vitro. It was developed by Kary Mullis in the 1980s. It is different from gene cloning. Rather than a series of manipulations involving living cells, PCR is carried out in a single test tube simply by mixing DNA with a set of reagents and placing the tube in a thermal cycler. Thermocycler is an equipment that enables the mixture to be incubated at a series of temperatures that are varied in a pre-programmed manner. The basic steps in a PCR experiment are: first, denaturation of template DNA. The mixture containing template DNA, oligonucleotide primers, and tagged DNA polymerase, a thermostable DNA polymerase, is heated to 94 degrees Celsius. At this temperature, the hydrogen bonds that hold together the two strands of the double-stranded DNA molecules are broken, causing the molecules to denature. Now we have the separated single-stranded DNA. Second, annealing of the oligonucleotide primers. The mixture is cooled down to 50 to 60 degrees Celsius. The two strands of each molecule could join back together at this temperature, but most do not, because the mixture contains a large number of short DNA molecules called oligonucleotides or primers, which anneal to the DNA molecules at specific positions. Third step, the synthesis of new DNA. For this, the temperature is raised to 74 degrees Celsius, which is the optimum working temperature of the tagged DNA polymerase. Tagged DNA polymerase attaches to one end of each primer and synthesizes new strands of DNA complementary to the template DNA molecule. Now we have four strands of DNA instead of the two that were at the beginning. Again, the temperature is increased back to 94 degrees Celsius. The double-stranded DNA molecule, each of which consists of one strand of the original molecule and one new strand of DNA, denatures into single strands. This is the second cycle of denaturation, annealing, synthesis, which gives 8 DNA strands. This cycle is repeated 25 to 30 times, which gives around 50 million new double-stranded molecules. Now, why gene cloning and PCR are so important? Even though these procedures are straightforward, both techniques can provide a pure sample of an individual gene separated from all the other genes in the cell. PCR can be completed in a few hours when compared to gene cloning, which requires maybe a week or a month to get the clone. Gene cloning can be used to isolate long genes, whereas in PCR only small fragments of DNA can be processed. Isolation and purification of DNA. The genetic engineers will at times need to prepare at least three distinct kinds of DNA. Total cell DNA, that is genomic DNA along with plasmids, pure plasmid DNA, and phage DNA. Preparation of total cell DNA. The procedure is divided into four stages. 1. Bacterial culture is grown and harvested. Second, cells are broken open to release their content. Third, cell extract treated to remove all components except DNA. And fourth, DNA solution is concentrated. Bacteria are grown in liquid media such as defined medium M9 and undefined medium Luria batani (LB broth). To prepare a cell extract, the bacteria must be obtained in small volumes. For example, OD at 600 nanometer must be 0.8 into 10 power 9 cells per milliliter. centrifuged at low speed to obtain the bacterial pellet. M9 medium contains sodium hydrogen phosphate, potassium dihydrogen phosphate, sodium chloride, ammonium chloride, magnesium sulfate, glucose, and calcium chloride. LB medium consists of tryptone, yeast extract, and sodium chloride. The bacterial culture is centrifuged at 8000 rpm for 10 minutes. The supernatant will be having the DNA and proteins, whereas the cell debris will form the pellet at the bottom of the centrifuge tube. Chemical lysis is used for cell disruption by using lysozyme, EDTA, ethylene diamine tetraacetate, or with a combination of both. Lysozyme digests polymeric compounds from cell wall which gives rigidity, and on the other hand EDTA removes magnesium ions from cell envelope and inhibits cellular enzymes that degrade DNA. STS, sodium dodecyl sulfate, is also added to remove liquid molecules. Purification of DNA from cell extract. One method is degradation with reagents. The standard way is the addition of phenol or 1:1 mixture of phenol and chloroform, which precipitates protein. But it has a disadvantage. The single extraction is not sufficient for purification. It requires repetition of the method, which will cause breakage of DNA molecule. A solution for this is the usage of protease enzymes, pronase or proteinase K, before phenol extraction. And to remove RNA, ribonuclease enzyme is used. Another method is ion exchange chromatography. It works on the principle separation of molecules based on their electrical charge. DNA, RNA, and some proteins are negatively charged, so the chromatographic matrix or resin used is positively charged. Molecules are eluted by increasing salt concentration of the solute. At a particular concentration, proteins and RNA will be eluted at first, which can be discarded. And at a different salt concentration, the DNA will be detached from the resin and can be collected. After purification, the next step is concentration of DNA. Organic extraction results in thick solution of DNA. Other purification methods gives dilute solutions which has to be concentrated. Ethanol precipitation is frequently used method. In the presence of salt, monovalent cations such as sodium ion, and at a temperature -20 degrees Celsius, absolute ethanol precipitates polymeric nucleic acids. With thick DNA solution, ethanol can be layered on top of the sample, causing molecules to precipitate at the interface. A glass rod can be pushed into the DNA solution, and when removed, DNA molecules adhere to it, which can be pulled out as a long fiber. If ethanol is mixed with dilute DNA solution, the precipitate can be collected by centrifugation and dissolved in water. Advantages: it leaves short chain and monomeric nucleic acid in solution. Ribonucleotides produced by ribonuclease treatment are lost. Measurement of DNA concentration. It is measured by ultraviolet absorption spectrophotometry. UV radiation absorbed by DNA solution is directly proportional to the amount of DNA in the sample. Absorbance is measured at 260 nanometer. An absorbance of 1 corresponds to 50 microgram of double-stranded DNA per milliliter. A pure DNA sample shows 1.8 absorbance at 260 and 280 nanometer. Ratio less than 1.8 indicates that the preparation is contaminated either with protein or phenol. Other methods. For example, if plant or animal cell DNA is to be purified, it requires some modifications in the process. The chemical used to disrupt a bacterial cell do not work with other organisms. For example, lysozyme has no effect on plant cells. The biochemical content of the cells vary. Plant cells contain large amount of carbohydrates which are not removed by phenol extraction. One method is the use of detergent cetyl trimethyl ammonium bromide (CTAB) forms an insoluble complex with nucleic acids. With plant cell extract, it precipitates nucleic acid CTAB complex. The precipitate is collected by centrifugation and resuspended in 1 molar sodium chloride. Nucleic acid concentrated by ethanol precipitation, and then the RNA are removed by ribonuclease treatment. The figure shows the CTAB method for purification of plant DNA. The CTAB forms a complex with a nucleic acid. When centrifuged, the complex will be pelleted out and the supernatant contains carbohydrates, proteins, etc. The pellet is resuspended in 1 molar sodium chloride and further the ethanol precipitation and ribonuclease treatment is carried out. Thus, we obtain a pure DNA. Another method is the use of guanidinium thiocyanate in ion exchange chromatography. It has two properties. Firstly, it denatures and dissolves all biochemicals other than the nucleic acids. Secondly, DNA binds tightly to silica particles in the presence of guanidinium thiocyanate. DNA can be obtained by washing the column with water.
Process of recombinant DNA technology. 1. Isolation of genetic material. The first step is the isolation of desired gene from a cell and the digestion of bacterial cell wall by enzymatic action. Process of recombinant DNA technology. 1. Isolation of genetic material. The first step is the isolation of desired gene from a cell and the digestion of bacterial cell wall by enzymatic action. This is achieved by treating the bacterial cells of plant or animal tissue with enzymes like lysozyme bacteria, cellulase, plant cells, and chitinase, fungus. Genes are located on DNA molecules centrifugation with proteins, such as histones. RNA can be removed by treatment with ribonuclease. Proteins can be removed by protease. Other molecules can be removed by appropriate treatments and further subjected to gradient centrifugation. Ultimately, purified DNA is precipitated out after the addition of chilled ethanol. 2. Cutting of DNA at specific locations. The purified DNA is cut into number of fragments by enzyme called restriction endonucleases. Each restriction endonuclease functions by inspecting the length of DNA and recognizes a specific palindromic nucleotide sequence in that DNA. Then it will bind to the DNA and cut each of the two strands of the double helix at specific points. These are called molecular scissors. They were first discovered by Nathans in 1970. Over 250 restriction enzymes have been isolated so far. When linear DNA is treated with restriction enzyme, a large number of DNA fragments are formed. The resultant fragments are separated by gel electrophoresis. Finally, the desired DNA fragments are selected by Southern blotting technique. 3. Amplification of gene of interest using PCR. Once the desired fragments of DNA genes are obtained, they are inserted into suitable vector to produce indefinite number of copies of genes. This is known as gene cloning. A cloning vector acts as a vehicle to carry the desired gene. An ideal cloning vector should have following properties. 1. It must have low molecular weight. 2. It must have unique cleavage site for the activity of restriction enzymes at single point. 3. It must be able to replicate truly inside host cell after its introduction. 4. It must contain genes which provide resistance to antibiotics. Several types of vectors are used in recombinant DNA technology like plasmids, phages, etc. The enzyme DNA polymerase helps in replication. If the process of replication of DNA is repeated many times, the DNA segment can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated replication is achieved by use of thermostable DNA polymerase, which remains active during high temperature-induced denaturation of double-stranded DNA. The amplified fragment of desired gene can be used to ligate with vector for further cloning. To isolate a plasmid, the bacterial cell is treated with EDTA, ethylene dimine tetraacetic acid, along with lysozyme enzyme to digest the cell wall. Then the bacterial cell subjected to centrifugation in sodium lauryl sulfate solution to separate the plasmid. The isolated plasmid DNA is cut with same restriction endonuclease enzyme. The enzymatic cleavage cuts circular plasmid into a linear molecule having sticky ends. The two sticky ends of this linear plasmid are joined to the ends of the desired gene. The enzyme DNA ligase joins the complementary ends of plasmid DNA with that of desired gene by covalent bonding to regenerate circular hybrid called recombinant rDNA or chimeric DNA. 4. Introduction of recombinant DNA into the host. The recombinant plasmid is transferred into a suitable bacterial host cell, generally E. coli, by a method known as transformation for the expression of desired gene. The cells into which the recombinant DNA are inserted are called transformed cells. Example, if rDNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin-resistant cells. Only transformants will grow on agar plates containing ampicillin. The transformed recipient cells will die. The ampicillin resistance gene is called selectable marker. Bacterial cell walls are not ordinarily permeable to such recombinant vectors, but reaping in dilute solution of calcium chloride renders the bacterial cell wall permeable to the recombinant vectors. Inside the host cell, this rDNA
Now let us see what are plasmids and how they are purified. A bacterial cell consists of three types of nucleic acid: chromosomal DNA, RNA, and plasmid. Plasmids are circular molecules of DNA that lead an independent existence in the bacterial cell. Plasmids almost always carry one or more genes, and often these genes are responsible for a useful characteristics displayed by the host bacterium. For example, the ability to survive in normally toxic concentrations such as chloramphenicol or ampicillin is often due to the presence in the bacterium of a plasmid carrying antibiotic resistance gene. Antibiotic resistance is often used as a selectable marker to ensure that bacteria in a culture contains a particular plasmid. They have at least one DNA sequence that can act as an organ of replication, so they are able to multiply within the cell quite independently of the bacterial chromosome. A few types of plasmids are also able to replicate by inserting themselves into the bacterial chromosome. These integrated plasmids or episomes may be stably maintained in this form through numerous cell divisions, but at some stages exist as independent elements. The size and copy number of a plasmid are particularly important for cloning. They range from 1 kilobase pair for the smallest to over 250 kilobase pair for the largest plasmids. The copy number refers to the number of molecules of an individual plasmid that are normally found in a single bacterial cell. Plasmid classification. Plasmids are classified into different types. First one, fertility or F plasmid, Tra gene for conjugal transfer. Second, resistance or R plasmid. Third, Col plasmid. It codes for colicin, protein that kill other bacteria. Next, degradative plasmid. It metabolizes unusual molecules such as 12 and salicylic acid present in the medium. Fifth, virulence plasmid confers pathogenicity on host bacterium. Episomes. Episomes are capable of existing outside of the chromosome. They are also designed to integrate into the chromosome following their movement from one cell to another. Now we'll look at the preparation of plasmid DNA. The process is same as that of the total cell DNA preparation. The only distinction is in the separation of plasmid DNA from the complete bacterial chromosomal DNA. It is essential as plasmids are used as cloning vectors. The isolation of pure plasmids are based on several physical differences such as size and the difference in conformation, that is, spatial configuration of the molecule. It may be linear and circular. For example, the largest plasmids are only 8% of the size of E. coli chromosome. The separation on the basis of size. Size fractionation is performed. The cell disruption must be carried out very gently to prevent whole breakage of bacterial DNA. The DNA fragments will be much larger, which can be centrifuged out with the cell debris. Treatment with EDTA and lysozyme is carried out in the presence of sucrose to prevent the cell from immediate bursting. Spheroplasts are formed, cells with partially degraded cell walls that retain an intact cytoplasmic membrane. The cell lysis induced with a non-ionic detergent Triton X100 which causes little DNA breakage, and further the clear lysate contains the plasmid DNA. Separation on the basis of conformation. Plasmids exist in the cell as supercoiled molecules. The supercoiling occurs because the double helix of the plasmid DNA is partially unwound during the plasmid replication process by enzyme called topoisomerases. The supercoiled conformation can be maintained only if both polynucleotide strands are intact, hence the name covalently closed circular DNA. If one of the polynucleotide strand is broken, the double helix reverts to its normal relaxed state called open circular DNA. Alkaline denaturation. This technique works on the principle that the non-supercoiled DNA is denatured at narrow pH range. If sodium hydroxide at pH 12 to 12.5 is added to a cell extract or cleared lysate, the hydrogen bonding in non-supercoiled DNA is broken. The double helix unwind and the polynucleotide chains are separated. If acid is added to this, these denatured strands aggregates into tangled mass which can be centrifuged, leaving plasmid DNA in the supernatant. Additional advantage is that most protein and RNA also become insoluble and can be removed by centrifugation step. So further purification steps are not needed. The next method is ethidium bromide cesium chloride density gradient centrifugation. A density gradient is produced by centrifuging a solution of cesium chloride at a very high speed. Macromolecules form bands at distinct points in the gradient. It depends on Boyne density. DNA has a Boyne density of 1.7 gram per centimeter cube, so it migrates to the point in the gradient where the cesium chloride density is 1.7 gram per centimeter cube. Protein has low density, so it floats on the top of the tube. RNA forms a pellet at the bottom. Density gradient centrifugation in the presence of ethidium bromide can be used to separate supercoiled DNA from non-supercoiled molecules. Ethidium bromide binds to DNA molecules by intercalating between adjacent base pairs, causing partial unwinding of the double helix. This unwinding decreases the Boyne density for linear DNA to 0.125 gram per centimeter cube, whereas supercoiled DNA has little freedom to unwind and can only bind a limited amount of ethidium bromide. The Boyne density of supercoiled DNA is about 0.085 gram per centimeter cube. It forms a band at different positions in the ethidium bromide cesium chloride gradient. The position of DNA bands can be seen by shining UV radiation. The ethidium bromide fluoresces. Plasmid DNA is removed by puncturing the side of the tube and withdrawing with a syringe. Ethidium bromide bound to plasmid DNA is extracted with N-butanol and cesium chloride removed by dialysis. The figures shows the intercalation of ethidium bromide and the process of extraction of supercoiled DNA. Next is plasmid amplification. The aim of amplification is to increase the copy number of plasmids. Multi-copy plasmids are able to replicate in the absence of protein synthesis. After a satisfactory cell density has been reached, a protein synthesis inhibitor, for example chloramphenicol, is added and the culture is incubated for 12 hours. Plasmid replicates whereas the chromosome replication and cell division is blocked. Thousands of copy number of plasmid may be attained.
Molecular cloning is a process of isolation of a specific DNA fragment and transfer of this fragment into a plasmid vector. As a part of a plasmid vector, the DNA fragment could be easily amplified, sequenced, stored for long periods of time, and used for gene expression and other functional studies. Two starting ingredients of molecular cloning are a plasmid vector and a DNA fragment that has to be inserted in it. The DNA fragment is usually a gene or other functional region from a living cell, or it could be an artificial sequence with properties useful for a researcher. Plasmid vector is a circular piece of DNA that could be easily amplified in E. coli, stored for long periods of time, and easily manipulated in a test tube. A typical plasmid vector contains an origin of replication that allows it to be replicated inside a bacterial cell, a selection marker, for example a beta-lactamase gene coding for ampicillin resistance, and a multiple cloning site which could be cleaved with several restriction enzymes such as BAMH1, ECAR1 or NCL1. In many vectors, the multiple cloning site is surrounded by sequences of promoter and terminator that guide expression of inserted genes after the vector is introduced inside a cell. To be used for molecular cloning, both vector and insert DNA are treated with restriction enzymes that cleave double-stranded DNA molecules, producing overhanging single-stranded nucleotide tails. After their ends have been prepared with restriction enzymes, vector and insert are combined together, and another enzyme called DNA ligase is added to the mix. And while complementary base pairing of single-stranded overhangs brings the ends of vector and insert together, the DNA ligase fuses them into one intact DNA molecule. In order to make multiple copies of this molecule, the ligation mix is introduced inside the E. coli cells in a process called transformation. During the transformation, the cell DNA mixture is kept on ice and then exposed to 42 degrees Celsius. A sudden change in temperature drives the DNA inside some of the E. coli cells. Then the cells are plated on a plate with growth medium, supplemented with a selective antibiotic. Only the cells that acquired the plasmid have resistance to the antibiotic and can grow on such a medium. After overnight incubation at 37 degrees, each transformed cell produces a colony of identical cells, oftentimes called a clone. The selected clones are then individually picked, grown even further in a liquid medium, and the DNA is extracted from them. So, in the process of molecular cloning, a DNA fragment that represented a tiny fraction of cell genome is integrated into a bacterial plasmid. As a part of the plasmid, this DNA fragment represents a quarter or more of total DNA in a test tube, and it could be effortlessly and endlessly amplified in E. coli. This presentation was brought to you by the
Isolation and purification of RNA. Ribonucleic acid is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. RNA and DNA are nucleic acids and along with lipids, proteins, and carbohydrates constitute the four major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, RNA is found in nature as a single strand folded onto itself rather than a paired double strand. Cellular organisms use messenger RNA to convey genetic information using the nitrogenous bases of guanine, uracil, adenine, and cytosine, denoted by the letters G, U, A, and C, and it directs synthesis of specific proteins. Many viruses encode their genetic information using an RNA genome. Some play an active role within cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes is protein synthesis, a universal function in which RNA molecules direct the synthesis of proteins on ribosomes. This process uses transfer RNA molecules to deliver amino acids to the ribosome, where ribosomal RNA then links amino acids together to form coded proteins. Three major types of techniques or combinations of them are employed in the isolation of nucleic acids: differential solubility, absorption methods, or density gradient centrifugation. A major goal of nucleic acid isolation is the removal of proteins, which is easily accomplished due to their different chemical properties. The highly charged phosphate backbone makes the nucleic acids rather hydrophilic as compared to proteins which are more hydrophobic. Hydrophilic means water-loving and hydrophobic means water-hating. But separating the different types of nucleic acids can be problematic due to their similar chemistries. Most nucleic acid isolation protocols involve a cell lysis step, enzymatic treatments, differential solubility, for example, phenol extraction or absorption to a solid support, and precipitation. Cell lysis. Nucleic acid must be solubilized from cells or other biological materials. They are carried out under denaturing conditions such as in the presence of STS, alkali, or boiling or characteristic agents. It promotes removal of proteins during subsequent steps and inhibits nucleases which will degrade the nucleic acids. The cell lysis and solubilization of RNA will typically be carried out in the guanidine salt, guanidine thiocyanate. Guanidine is a strong characteristic agent and will inhibit RNase. The strong denaturing effect of this salt will also promote better phenol extraction. Enzymatic treatment. It degrades unwanted components. For example, inclusion of proteases, proteinase K, in the lysate will promote the removal of protein. Proteinase K is still active at 55 degrees Celsius in the presence of 0.5% STS. The elevated temperature and STS improve solubility and inhibit any DNase activity that may be present in the lysate. Nucleases can also be used to remove unwanted nucleic acids. Phenol extraction. Phenol is an organic solvent used to separate proteins from nucleic acids. Proteins are hydrophobic and partitions in the organic phase. Nucleic acids are highly charged and partitions in the aqueous phase. Advantages of these methods are it is easy to carry out and adapted to many applications. It is applied over a wide range of volume. It is widely used for isolation of high molecular weight genomic DNA. Phenol extraction is accomplished by mixing the sample with an equal volume of phenol, which has been previously saturated with a tris buffer at a pH 8 containing EDTA and NaCl. Phenol should be molecular biology grade and stored at -20 degrees Celsius. The saturated solution is stored at 4 degrees Celsius. The two phases are completely mixed by vortexing or gently mixing and separated by centrifugation. The upper aqueous phase, which contains the nucleic acids, is retained. Proteins will be visible as flocculent material at the top of the phenol phase. The two phases need to be carefully separated. The aqueous phase can be re-extracted with phenol to remove more proteins. Phenol is hazardous that needs to be disposed of properly. A common variation of phenol extraction is a mixture of phenol, chloroform, isoamyl alcohol in the ratio 25:24:1. The more organic chloroform removes lipids, denatures more protein and mixes less with the aqueous phase, leading to more efficient extraction. For RNase isolation, precautions against RNase activity must be taken. RNase is an extremely stable and active enzyme. Gloves should be worn at all times and sterile plastic ware should be used whenever possible to avoid exogenous RNase to the sample. Glassware needs to be treated with DEPC water, that is diethyl pyrocarbonate or diethyl dicarbonate water, and autoclave to inactivate any RNase. Buffers should be prepared from DEPC water or RNase inhibitors included. In case of RNA, the phenol is saturated with a buffer of pH 4 to 6. It involves phase separation by addition and centrifugation of a mixture of solution containing phenol, chloroform and a characteristic agent guanidinium thiocyanate and aqueous sample. Guanidinium thiocyanate results in the denaturation of proteins and RNAs, separating RNA from ribosomes. Addition of chloroform forms a colorless upper aqueous phase containing RNA, an interphase containing DNA, and a lower phenol chloroform phase containing protein. RNA is collected from the upper aqueous phase by alcohol precipitation followed by rehydration. One of the advantages of this method is the stabilization of RNA and rapid denaturation of nucleases. Besides the advantages, it has several drawbacks, such as it is difficult to automate, needs labor and manual intensive processing, and use of chlorinated organic reagents. The low pH will result in DNA partitioning in the lower phase. If DNA contamination is a problem, it is possible to purchase RNA-free DNAs. Long-term storage is best as a precipitate in 70% ethanol at -20 degrees Celsius. The figure shows the traditional phenol extraction of RNA and DNA. Here, the organic phase contains the protein and DNA is in between, that is in the interphase, and RNA is in the aqueous phase. This is at the pH 4, and at pH 8 the protein is in the organic phase and both the DNA and RNA are present in the aqueous phase. Precipitation of RNA with lithium chloride. Lithium chloride selectively precipitate RNA. Large RNA such as rRNA and mRNA are insoluble at high ionic strength, whereas small RNA such as tRNA and 5S rRNA and DNA generally remain soluble. Following either phenol or guanidine extraction, an equal volume of 8 molar lithium chloride is added. The sample is mixed vigorously and incubated at -20 degrees Celsius. The precipitate is collected by centrifugation and reprecipitated if necessary. Affinity chromatography. Eukaryotic mRNA contains a stretch of A residues, that is adenine residues, at its 3' end, which is added post-transcriptionally. It is possible to isolate mRNA by affinity chromatography on oligo-DT columns. The RNA solution is passed over an oligo-DT column under conditions which promote base pairing. Only RNA with a poly-A tail binds. The poly-A RNA is eluted under conditions usually low salt and high temperature which break the base pairing. The figure shows the isolation of mRNA using oligo-DT column chromatography. The mRNA with poly-A tail binds to the oligo-DT matrix and can be extracted with low salt buffer. Now we know how to isolate and purify different nucleic acids such as DNA, RNA, and plasmids. The next topic is restriction enzymes. Let us look into some history which led to the discovery of restriction enzymes. In the early 1950s, experiments by two teams of researchers, Salvador Luria working with Mary Human and Joe Bertani working with Jean Weigel, showed that some strains of bacteria were more resistant to viral infections than others. Viruses that infect bacterial cells are known as bacteriophages. The main goal of these researchers were to produce more bacteriophages by infecting their genome into a bacterial host cell, that is, by using the host cell machinery to copy their genome and expressing bacteriophage genes. However, the researchers found that some bacterial strains appeared to be less vulnerable to bacteriophage infections and resisted the hijacking of their cell machinery by bacteriophages. When they had a deeper look into the self-defense mechanisms of these bacteriophage-resistant bacteria, it revealed that a group of enzymes called restriction endonucleases or restriction enzymes was used as their secret weapon. These enzymes later opened the path to a powerful research tool to sequence genomes and also to create the first synthetic cell. These were the two scientific research milestones in the 1950s. The discovery of restriction enzymes began with a hypothesis. In the 1960s, Werner Arber observed a dramatic change in the bacteriophage DNA after it invaded these resistant strains of bacteria. It was degraded and cut into pieces. He posited that the bacterial cells expressed a specific enzyme that degrades only bacteriophage DNA and not its own DNA. Then a question arose: how would a DNA degrading enzyme distinguish between the two, that is, the phage DNA and the bacterial DNA? Arber hypothesized bacterial cells might express two types of enzymes: a restriction enzyme, which recognizes and cuts up the foreign bacteriophage DNA, and a modification enzyme, which recognizes and modifies the bacterial DNA to protect it from the DNA degrading activity of its very own restriction enzyme. He predicted that the restriction enzyme and the modification enzyme act on the same DNA sequence called a recognition sequence. This prediction was confirmed in the late 1960s by Stuart Lin and Arber when they isolated a modification enzyme called methylase and a restriction enzyme responsible for bacteriophage resistance in the bacterium Escherichia coli. The methylase enzyme added protective methyl groups to DNA, and the restriction enzyme cut unmethylated or unprotected DNA at multiple locations along its length. In 1970, Hamilton Smith not only independently verified Arber's hypothesis but also elaborated on the initial discovery by Lin and Arber. He successfully purified a restriction enzyme from another bacterium, Haemophilus influenzae, and definitively showed that this enzyme cut DNA in the center of a specific 6 base pair sequence. Interestingly, he also showed that this enzyme did not cut at this very same DNA sequence when it occurred in Haemophilus influenzae host cell DNA. 1 year later, in experiments carried out by Dan Nathans and Kathleen Dana, restriction enzymes were used as scientific research tools. They used Smith's restriction enzyme to cut the 5000 base pair genome of the SV40 virus, which infects monkey and human cells, and identified 11 differently sized pieces of DNA. When the SV40 genome was digested with different combinations of restriction enzymes, the sizes of the resulting pieces of DNA could be used to deduce a physical map of the SV40 viral genome, a groundbreaking method for inferring gene sequence information. This method of cutting a DNA molecule into smaller pieces is called a restriction enzyme digest, and it quickly became a powerful tool for generating physical maps of a multitude of genomes which at the time was a precious revelation in the early stages of genome sequencing. For this groundbreaking set of discoveries, Arbor, Smith, and Nathans were jointly awarded the Nobel Prize in Physiology or Medicine in 1978. Restriction enzymes are also known as molecular scissors. Different bacterial strains express different restriction enzymes, allowing them to balance their own genes against those of invading bacteriophages. The known variety of restriction enzymes is staggering. To date, more than 4,000 different restriction enzymes that collectively recognize more than 360 different recognition sequences have been isolated from a wide variety of bacterial strains. Based on DNA sequence analysis, scientists know that there are many more restriction enzymes out there waiting to be characterized. The recognition sequences of these enzymes are typically 4 to 6 base pair in length, and they are usually palindromic, which means that their restriction sequence reads the same in the 5' to 3' direction on both DNA strands. Restriction endonucleases. Gene cloning requires the DNA molecules to be cut in a very precise and reproducible manner. Each vector must be cleaved at a single position to open up the circle so that the new DNA can be inserted. A molecule which is cut more than once is of no use as a cloning vector. DNA to be cloned must also be cleaved because firstly, if the aim is to clone a single gene which is 2 to 3 kilobase pairs, the large DNA molecule should be cut. Secondly, large DNA molecules may have to be broken down simply to produce fragments small enough to be carried by the vector. For example, M13-based vector are inefficient at cloning DNA molecule more than 3 kilobase pairs in length. The discovery of restriction endonucleases was made in early 1950s when it was shown that some bacterial strains are immune to bacteriophage infection, a phenomenon called as host-controlled restriction. Restriction occurs because the bacterium produces an enzyme that degrades the phage DNA before it has time to replicate and synthesize new phage particles. The destruction of bacterial DNA is prevented because it carries additional methyl group that block the degradative enzyme action. Restriction endonucleases are classified into three different classes. The types 1 and 3, which are rather complex and have a limited role in genetic engineering. Type 2 restriction endonucleases are the cutting enzymes which are very important in gene cloning. Type 2 restriction endonucleases cut DNA at specific nucleotide sequences. Each endonuclease has a specific recognition sequence at which it cuts a DNA molecule. The figure shows the restriction enzymes and their recognition sequences. For example, PUV1 isolated from Proteus vulgaris cuts DNA only at the hexanucleotide CGA-TCG. PUV2 cuts at a different hexanucleotide CAGC-TG. Many restriction endonucleases recognize hexanucleotide target sites, but others cut a 4, 5, 8, or even longer nucleotide sequences. SAU3A recognize CATC and ALU1 cuts at AGCT. There are also examples of recognition sequences, meaning that they cut DNA at any one of a family of related sites. HINF1, for instance, recognizes GANTC So cuts at GAATC, GATTC, GAGTC and GACTC. Classification of restriction endonucleases. Type 1 endonucleases are a complex of three subunits with methylase activities. It is ATP dependent. It cut both strands at a non-specific location around 1000 base pairs away from recognition site. They are less abundant than type 2. Examples are EcoK1, EcoA1, CFRA1. Type 2 endonucleases are homodimers. Methylase are separate and are single subunit enzymes. They are magnesium ion dependent. They recognize symmetric DNA sequences and cleave within sequences. They are most common enzymes, about 93%. Examples are EcoR1, BAMH1, HIND3. Type 3 endonuclease and methylase are separate two subunit complexes with one subunit in common. It is ATP dependent. It cleaves only one strand, 24 to 26 base pair downstream of the 3' recognition site. They are rare type of endonuclease. For example, EcoP1, HINF3, EcoP15-1. Action. Restriction endonucleases scan the length of the DNA, binds to the DNA molecules when it recognizes a specific sequence, and make one cut in each of the sugar phosphate backbones of the double helix by hydrolyzing the phosphodiester bond. Specifically, the bond between the 3' oxygen atom and the phosphate atom is broken. 3' OH and 5' PO4 3- is produced. Magnesium ion is required for the catalytic activity of the enzyme. It holds the water molecule in position where it can attack the phosphoryl group. End products of restriction enzyme. Three types of end products are produced by the type 2 endonucleases: 3' overhang, 5' overhang, and blunt end. Many endonucleases make a simple double-stranded cut in the middle of a recognition sequence, resulting in blunt end or flush end. PUV2 and ALU1 are blunt end cutters. Other endonucleases does not cut the DNA at exact same position. The cleavage is staggered by 2 or 4 nucleotides, resulting short single-stranded overhangs at each end. They are called sticky or cohesive ends. The figures show 5' overhang of EcoR1 and 3' overhang of PST1. Frequency of recognition sequences. The number of recognition sequences can be calculated mathematically. For example, a tetranucleotide sequence GATC should occur once every 4^4, that is 256 nucleotides, and hexanucleotide GGATCC occur every 4^6, that is 4096 nucleotides. An assumption is that the GC content will be equal to 50%, but in practice, these assumptions are not valid. For example, the lambda DNA at 49 kb should contain 19 sites for restriction with hexanucleotide, but occurs less frequently. The figure shows the cleavage site on lambda DNA and the fragment size. From this figure, we can understand that the restriction sites does not occur at definite intervals. Restriction digestion in laboratory. As an example, we will consider how to digest a sample of lambda DNA with a concentration of 125 microgram per milliliter with BGL2. First of all, the required amount of DNA must be pipetted into a test tube. The amount of DNA that will be restricted depends on the nature of the experiment. In this case, we shall digest 2 microgram of lambda DNA which is contained in 16 microliter of the sample. Very accurate micropipettes will therefore be needed. The other main component in the reaction will be the restriction endonuclease. Before adding the enzyme, the solution containing the DNA must be adjusted to provide the correct conditions to ensure maximal activity of the enzyme. Most restriction endonucleases function adequately at pH 7.4, but different enzymes vary in their requirements for ionic strength, usually provided by sodium chloride and magnesium concentration. It is also advisable to add a reducing agent such as dithiotritol (DTT), which stabilizes the enzyme and prevents its inactivation. Providing the right conditions for the enzyme is very important. Incorrect sodium chloride or magnesium ion concentration may not only decrease the activity of the restriction endonuclease but may also cause change in the specificity of the enzyme so that DNA cleavage occurs at additional non-standard recognition sequences. The buffer used contains tris-HCl, magnesium chloride, sodium chloride, and DTT. In this example, a suitable final volume for the reaction mixture would be 20 microliter. So we add 2 microliter of 10x BgL2 buffer to the 16 microliter of DNA already present. The restriction endonuclease can now be added. By convention, 1 unit of enzyme is defined as the quantity needed to cut 1 microgram of DNA in 1 hour. So we need 2 units of BgL2 to cut 2 microgram of lambda DNA. BgL2 is frequently obtained at a concentration of 4 units per microliter. So 0.5 microliter is sufficient to cleave the DNA. The final ingredients in the reaction mixture are therefore 0.5 microliter BgL2 plus 1.5 microliter water, giving a final volume of 20 microliter. The last factor to consider is incubation temperature. Most restriction endonucleases work best at 37 degrees Celsius, but few have different requirements. For example, Taq polymerase I must be incubated at 65 degrees Celsius to obtain maximum activity. After 1 hour, the restriction should be complete. If the DNA fragments produced by restriction are to be used in cloning, the enzyme must be destroyed so that it does not accidentally digest other DNA molecules that may be added at a later stage. For many, a short incubation at 70 degrees Celsius is sufficient. For others, phenol extraction or addition of EDTA, which binds magnesium ions, preventing restriction enzyme action, is used. And now, how do we analyze the result of restriction endonuclease cleavage? A restriction digest results in a number of DNA fragments, the size of which depends on the exact positions of the recognition sequences for the endonuclease in the original molecule. Whether a DNA molecule is cut or not can be determined by testing the viscosity of the solution. Larger DNA molecules result in a more viscous solution than smaller ones. However, working out the number and sizes of individual cleavage products was possible after the development of gel electrophoresis. So, separation of molecules by gel electrophoresis. Electrophoresis, like ion exchange chromatography, is a technique that uses differences in electrical charge to separate the molecules in a mixture. DNA molecules have negative charges, so when placed in an electric field, they migrate towards the positive pole. The rate of migration of a molecule depends on two factors, its shape and its charge to mass ratio. In gel electrophoresis, a gel is usually made of agarose polyacrylamide, or a mixture of the two, comprises a complex network of pores through which the DNA molecule must travel to reach the positive electrode. The smaller the DNA, the faster it can migrate through the gel. Gel electrophoresis separates DNA molecules according to their size. The composition of gel determines the sizes of the DNA molecules that can be separated. A 0.5 cm thick slab of 0.5% agarose, which has relatively large pores, would be used for molecules in the size range 1 to 30 kb, allowing, for example, molecules of 10 kb and 12 kb to be clearly distinguished. A very thin, that is 0.3 mm, that is 40% polyacrylamide gel, can be used to separate a much smaller DNA molecule in the range 1 to 300 base pair and could distinguish molecules differing in length by just a single nucleotide. Now we have separated the DNA fragments using gel electrophoresis. And how do we visualize those bands? So the easiest way to see the results of a gel electrophoresis is to stain the gel with a compound that makes the DNA visible. Ethidium bromide is routinely used to stain DNA in agarose and polyacrylamide gels. Bands showing the positions of the different sizes of DNA fragments are clearly visible under ultraviolet irradiation after ethidium bromide staining. A non-mutagenic dye that stains DNA, green or blue, is used in many laboratories due to the hazardous nature of ethidium bromide and UV rays. The one drawback in staining is that there is a limit to its sensitivity. If less than 10 nanogram of DNA is present per band, it may not show the band after staining. So for small amounts of DNA, autoradiography is used. If the DNA is labeled before electrophoresis by incorporation of a radioactive marker into the individual molecule, then the DNA can be visualized by placing an X-ray sensitive photographic film over the gel. The radioactive DNA exposes the film, revealing the banding pattern. A DNA molecule is usually labeled by incorporating nucleotides that carry a radioactive isotope of phosphorus P32. Several methods are available, the most popular being nick translation and end filling. Nick translation refers to the activity of DNA polymerase I. Most purified samples of DNA contain some nicked molecules. However, carefully the preparation has been carried out, which means that DNA polymerase I is able to attach to the DNA and catalyze a strand replacement reaction. This reaction requires a supply of nucleotide. If one of these is radioactively labeled, the DNA molecule will itself become labeled. So nick translation can be used to label any DNA molecule, but may under some circumstances cause DNA cleavage. End filling is a gentler method that rarely causes a breakage of DNA, but unfortunately can only be used to label DNA molecules that have sticky ends. The enzyme used is the cleaner fragment, which fill in a sticky end by synthesizing the complementary strand. If the end filling reaction is carried out in the presence of labeled nucleotide, the DNA itself becomes labeled. Both the methods can be used for labeling DNA at very small quantities and can be detected in gels by autoradiography.
Endonucleases cleave DNA internally and do not require free DNA ends for activity. This is in contrast to exonucleases, which require and act on free DNA ends. In vivo, many endonucleases are involved in the repair of damaged DNA. Many of these enzymes have been adapted into workflows such as gene assembly and directional RNA-seq. Here, we will be highlighting two types of endonucleases, structure-independent and structure-dependent. Structure-independent endos randomly cleave internal phosphodiester bonds and are named such because they do not require a specific DNA structure for activity. They act on canonical single and double-stranded DNA. Structure-independent endos are primarily used to clean up DNA contamination. These are processive enzymes, and once activity has been initiated, the endo will continue to cleave DNA. Examples of structure-independent endos active on double-stranded DNA are micrococcal nuclease and DNase 1. An example of a structure-independent endo active on single-stranded DNA is mungbean nuclease. Structure-dependent endos, which include a wide variety of DNA repair enzymes, require a specific DNA structure or damaged base for activity, and will cleave an internal phosphodiester bond at that structure or damaged base. Apurinic or apyrimidinic sites, also known as AP or abasic sites, are common and occur spontaneously in DNA when a base is lost. This can be detrimental to the cell. An example of an endonuclease that will act on an AP site is Endo4. It catalyzes the cleavage of the DNA phosphodiester backbone at AP sites via hydrolysis, leaving a 1 nucleotide gap with 3' hydroxyl and 5' deoxyribose phosphate termini. 8-oxoguanine, more commonly known as 8-oxo-G, is a common DNA error that results from oxidizing guanine. This is also detrimental to the cell. An example of a DNA repair enzyme that removes this type of error is FPG. FPG is a bifunctional DNA glycosylase with DNA N-glycosylase and AP lyase activities. N-glycosylase activity releases damaged purines, including 2,6-diamino, 4-hydroxy-5-formamidopyrimidine, and 8-oxo-7,8-dihydroguanine, generating an AP site. The AP lyase activity of FPG cleaves an AP site via beta and delta elimination, creating a 1 nucleotide DNA gap with 5' and 3' phosphate termini. Some enzymes act only as glycosylases, that is, they only recognize and remove the damaged base but do not specifically cleave the DNA backbone. An example of this type of enzyme is UDG, which recognizes deoxyuridine, or DU, and removes it and leaves an AP site. To cleave the DNA backbone, another enzyme, such as endonuclease 8, is required. User enzyme contains a mixture of these two enzymes so that the damage is completely removed and the AP site is cleaved. Aside from DNA repair enzymes, there are also structure-specific endonucleases that act on DNA secondary structure. Thermostable Fen1, for example, recognizes a specific flap structure and cleaves this flap, leaving a 5' phosphate, which can then be ligated with a DNA ligase. For more information regarding NEB endonucleases,
Now we come to exonucleases. Exonucleases are enzymes that work by cleaving nucleotides one at a time from the end of a polynucleotide chain. A hydrolyzing reaction that breaks phosphodiester bond at either the 3' or the 5' end occurs. Eukaryotes and prokaryotes have three types of exonucleases involved in the normal turnover of mRNA. 5' to 3' exonuclease, which is a dependent decapping protein, 3' to 5' exonuclease, an independent protein, and poly A, specific 3' to 5' exonuclease. Significance to polymerase. RNA polymerase II is known to be in effect during transcriptional termination. It works with a 5' exonuclease to degrade the newly formed transcript downstream, leaving the polyadenylation site and simultaneously shooting the polymerase. This process involves the nucleases catching up the Pol II and terminating the transcription. Polymerase I then synthesizes DNA nucleotide in place of the RNA primer it had just removed. DNA polymerase I also has 3' to 5' and 5' to 3' exonuclease activity, which is used in editing and proofreading DNA for errors. The 3' to 5' can only remove one mononucleotide at a time, and the 5' to 3' activity can remove mononucleotides or up to 10 nucleotides at a time. In 1971, Lehman discovered nuclease in E. coli. Since that time, there has been numerous discoveries including exonuclease 2 to 8. Each type of exonuclease has a specific type of function or requirement. Exonuclease 1 breaks apart single-stranded DNA in a 3' to 5' direction, releasing deoxynucleoside 5' monophosphates one after another. It does not cleave DNA strands without terminal 3' OH groups because they are blocked by phosphoryl or acetyl groups. Exonuclease 2 is associated with DNA polymerase I, which contain a 5' exonuclease that clips off the RNA primer contained immediately upstream from the site of DNA synthesis in a 5' to 3' manner. Exonuclease 3 has four catalytic activities: 3' to 5' exodeoxyribonuclease activity, which is specific for double-stranded DNA, RNase activity, and 3' phosphate activity, AP endonuclease activity, which was later called endonuclease 2. Exonuclease 4 adds a water molecule so it can break the bond of an oligonucleotide to nucleoside 5 monophosphate. This exonuclease requires magnesium ion in order to function and works at higher temperatures than exonuclease 1. Exonuclease 5 is a 3' to 5' hydrolyzing enzyme that catalyzes linear double-stranded DNA and single-stranded DNA, which requires calcium ion. This enzyme is extremely important in the process of homologous recombination. And exonuclease 8 is 5' to 3' dimeric protein that does not require ATP or any gaps or nicks in the strand, but requires a free 5' OH group to carry out its function.
Exonucleases are enzymes that catalyze the removal of nucleotides in either the 5' to 3' or the 3' to 5' direction from the ends of single-stranded and/or double-stranded DNA. Removal of nucleotides is achieved by cleavage of phosphodiester bonds via hydrolysis. These important enzymes can have slightly different activities and therefore can be used for a wide variety of applications. Most exonucleases digest at nicks in the DNA. Some exonucleases remove one base at a time. Lambda exonuclease is an example of this and transforms double-stranded DNA into single-stranded DNA by chewing from the free end containing a 5' phosphate, degrading one strand preferentially but not the other. Other examples are EXO1 and EXO3. Other exonucleases, such as T5, EXO5, or EXO7, remove short oligos. The products of T5-exo and EXO5 digestion also include individual bases. Exonucleases, such as EXO7 and 5, digest in both the 5' to 3' and the 3' to 5' direction, while others, such as XOT and XO1, only work in one direction. Some exonucleases, such as XO1 and XOT, only digest single-stranded DNA while leaving behind double-stranded DNA. This can be useful to remove PCR primers, for example. Exonucleases, such as T7XO, digest only double-stranded DNA, while others, such as T5XO and XO5, can digest both single and double-stranded DNA. A unique enzyme is XO5, also known as RecBCD. This enzyme consists of 3 subunits. This includes helicase activity that unwinds double-stranded DNA to expose single-stranded DNA, as well as single-stranded exo and endonuclease activity, which digests the DNA. The end result is activity like an exonuclease, for example, shortening from the end. This enzyme is often used to clean up plasmid DNA because it selectively degrades linear DNA without touching circular DNA. As you can see, exonucleases work in many different ways and can be used individually or combined to achieve the activity that you require for your specific application. For more information regarding NEB exonuclease,
Ligases. Ligases are enzymes that join the nucleic acid molecules together. They can join both DNA and RNA. DNA ligase catalyzes the formation of a phosphodiester bond between the 5' phosphate of one strand and the 3' hydroxyl group of another. DNA ligases are magnesium ion dependent enzymes. The most widely used DNA ligase is derived from the bacteriophage T4. DNA ligase repair single-strand breaks or nick. Blunt-ended and sticky-ended ligation. Ligation reactions may be blunt-ended or sticky-ended. Sticky ends increase the efficiency of ligation. Types of DNA ligase. There are 2 families of DNA ligases, ATP dependent, which is found in eukaryotes, and NAD+ dependent, which are found in prokaryotes. In mammals, there are 4 types, 1 to 4, of DNA ligases. T4 DNA ligase. It is isolated from bacteriophage T4. It will ligate the ends of duplex DNA or RNA. This enzyme will join blunt end termini as well as ends with cohesive overhanging ends. This enzyme will also repair single-stranded nicks in duplex DNA, RNA or DNA-RNA duplexes. It requires ATP as a cofactor. Taq DNA ligase. This ligase will catalyze a phosphodiester bond between 2 adjacent oligonucleotides which are hybridized to a complementary DNA strand. The ligation is efficient only if the oligonucleotides hybridize perfectly with the template strand. The enzyme is active at relatively high temperature, that is 45 to 65 degrees Celsius. It requires NAD+ as a cofactor. T4 RNA ligase. It will catalyze formation of a phosphodiester bond between RNA-RNA oligonucleotides, RNA-DNA oligonucleotides, or DNA-DNA oligonucleotides. It requires ATP as a cofactor. This enzyme does not require a template strand. T4 RNA ligase can be used for a variety of purposes, including constructing RNA-DNA hybrid molecules.
DNA ligation is the formation of a covalent bond between adjacent DNA fragments, frequently a vector and a gene of interest. Ligation is usually the final step before transformation in the cloning workflow. DNA ligase may be used to join double-stranded DNA fragments with either blunt or cohesive ends to form recombinant DNA plasmids. A DNA ligase catalyzes the formation of a phosphodiester bond between juxtaposed 5' phosphate and 3' hydroxyl termini in duplex DNA. Ligation proceeds in 3 steps. Initially, the ligase is self-adenylated by reaction with free ATP or NAD. Next, the adenyl group is transferred to the 5' phosphorylated end of the donor strand. Lastly, the formation of the phosphodiester bond proceeds through the reaction of the adenylated donor end with the adjacent 3' hydroxyl acceptor, accompanied by the release of AMP. While T4 DNA ligase is the most commonly used ligase for cloning, there are several options from NEB to choose from, including convenient master mix formats. For help with selection of a DNA ligase optimized for a specific end type and application, view our selection chart at nebsticktogether.com. Visit Clone with NEB.
Methylases. It is also known as methyltransferase, found in bacteria to mammals. Usually, organisms that make restriction enzymes also synthesize DNA methyltransferase that protects their own DNA from cleavage. These enzymes recognize the same DNA sequence as the restriction enzyme they accompany. Methylation is the process of addition of methyl groups to adenine or cytosine bases within the recognition site and thereby modifying the site and prevent the DNA restriction. The recognition sequences of the methylases are the same as the associated endonucleases. Example, EcoR1 methylase recognizes and methylates at the sequence GAATTC. All methylases transfer the methyl group from S-adenosylmethionine (SAM) to a specific base in the recognition sequence, and SAM is a required component in the methylation reaction. Methylation of DNA usually has the effect of protecting the DNA from the related restriction endonuclease. However, there are methylases with minimal specificity. For example, SSS1 methylase will methylate cytosine residues in the sequence 5' CG3'. In this case, the methylated DNA will be protected from a wide variety of restriction endonucleases. Some restriction endonucleases will only cut DNA at their recognition sites if the DNA is methylated. For example, DPN1. Still, other restriction endonucleases will cut both methylated and non-methylated DNA at their recognition sequences. Example, BAMH1. DNA polymerases. All DNA polymerases share two general characteristics. They add nucleotides to the 3' OH end of a primer. The order of the nucleotides in the nascent polynucleotide is template directed. In addition to the 5' to 3' polymerase activity, polymerases can contain exonuclease activity. This exonuclease activity can proceed either in the 5' to 3' direction or in the 3' to 5' direction. Exonuclease activity in the 3' to 5' direction allows polymerase to correct a mistake if it incorporates an incorrect nucleotide, so-called error correction activity. It can also slowly degrade the 3' end of the primer. Exonuclease activity in the 5' to 3' direction will allow it to degrade any other hybridized primer it may encounter. Without 5' to 3' exonuclease activity, obstructing primers may or may not be physically displaced depending on the polymerase being used. Different polymerases have differing error rates of misincorporation and different rates of polymerization. Uses of polymerases. The various activities of the different polymerases lend them to a variety of applications. For example, restriction endonuclease can yield fragments of DNA with either 3' or 5' nucleotide overhangs. In the case of 5' overhangs, the 5' to 3' polymerase activity can fill these in to make blunt ends. In the case of 3' overhangs, the 3' to 5' exonuclease activity present in some polymerases, especially T4 DNA polymerase, can chew back these ends to also make blunt-ended DNA fragments. Nick translation. This method is used to obtain highly radiolabeled single-strand DNA fragments which make use of 5' to 3' exonuclease activity present in some polymerases, for example, E. coli DNA polymerase I. In this method, a DNA duplex of interest is nicked, that is, one of the strands is cut. Then DNA polymerase I is added along with radio-labeled nucleotides. The 5' to 3' exonuclease activity chews away the 5' end at the nick site and the polymerase activity incorporates the radio-labeled nucleotides. The resulting polynucleotide will be highly radio-labeled and will hybridize the DNA sequence of interest. Thermostable polymerases have the ability to remain functional at temperature ranges where the DNA duplex will actually melt and become separated. This has allowed the development of the polymerase chain reaction technique, which has had a profound impact on modern biotechnology. The incorporation of dideoxybases, that is, no hydroxyl groups on either the 2' or 3' carbon of the ribose sugar, led to termination of polymerase reaction. However, this chain termination by incorporation of dideoxynucleotides is the basis of the Sanger method of DNA sequencing as well as therapies to try to inhibit viral replication. T4 polynucleotide kinase. It catalyzes the transfer and exchange of a phosphate group from the G position of R-ATP to the 5' hydroxyl terminus of double-stranded and single-stranded DNA or RNA and nucleoside 3' phosphates. The enzyme will also remove 3' phosphoryl groups. Oligonucleotides which are obtained from automated synthesizers lack a 5' phosphate group and thus cannot be ligated to other polynucleotides. T4 polynucleotide kinase can be used to phosphorylate the 5' end of such polynucleotides. Points to remember. In this unit, you have learned about what is recombinant DNA technology, how a recombinant DNA is produced, what are the processes involved in the recombinant DNA technology, and which are all the enzymes involved in the process. So recombinant DNA molecule is a molecule of DNA from two different species that are inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. The processes which are involved in the recombinant DNA technology is isolation of DNA, fragmentation of DNA, amplification, and insertion of recombinant DNA into the host. Amplification can be done by PCR or by undergoing amplification inside the bacterial cell itself. And the restriction enzymes involved are endonucleases and exonucleases. Endonucleases will cleave the DNA fragment in between. whereas the exonucleases will cleave the nucleotides from the ends of the DNA fragment. The ligases will join the two different DNA fragments, whereas the methylases will add a methyl group to the DNA fragment so that it will be protected from the restriction enzymes. So these are some of the points which you have to remember in this unit. Some reference books are given here. T.A. Brown, An Introduction to Gene Cloning. It will be a very useful book for studying about gene cloning and the different enzymes and vectors used. And Dubé, that is also a very good book. You can refer both the books. In the next session, you will be learning about vectors, different types of vectors, prokaryotic vectors and eukaryotic vectors. In prokaryotic vectors, you will be learning about natural as well as artificial vectors. There are a number of vectors constructed by many scientists, and also eukaryotic vectors, the vectors which are useful for the plants and animals. Thank you for listening.